Hey everyone, Dr. D here, and in this video we're going to be finishing off Chapter 4, so let's get to it. Alright, so here where we left off, we were about to talk about the Golgi apparatus and its structure and function. So let's go ahead and start. The Golgi apparatus consists of flattened, let me maximize this, flattened membranous sacs called cisternae, or Golgi bodies. Let me show you what it looks like. Remember this slide here? This is a typical animal cell. So where are these membranous sacs? It's right here. This is the Golgi apparatus. Notice how these are all membranes. The Golgi is made out of a bunch of membranes. These are the membranous sacs that they're talking about, membranous sacs. So what you say is that there are a stack of membranous sacs, which is the Golgi. And the job of the Golgi is to receive proteins, modify proteins, and then ship them to where they need to go. That's the job. So it's a shipping and processing center for proteins. But how does it get those proteins? Where do the proteins come from? Well, let me show you. Do you remember last time in the previous video I told you that proteins are made at the rough ER, and those proteins can be membrane proteins or proteins that float around in the lumen of the ER, and then that ER membrane can pinch until it pinches off as a vesicle, a transport vesicle. Well, let me show you. See, here are some of those membrane proteins inside of the rough ER, and the membrane proteins are actually part of the membrane of the rough ER, and then there's other proteins inside. And what happens? Those proteins leave the ER in a transport vesicle. Basically, the ER membrane can pinch until it pinches off as a vesicle. That vesicle contains those proteins, membrane proteins and other. Those proteins make their way to the Golgi. And do you remember what I said about the membranes inside of the cell? The membranes are basically like soap bubbles. So you know how soap bubbles work. Two soap bubbles. If two soap bubbles touch, they can form one big soap bubble. If you mess with a big soap bubble, that can break off into two smaller soap bubbles. You get the idea. So what's happening here is the ER membrane pinches until it pinches off a soap bubble, okay? And it's not really a soap bubble, obviously. It's a phospholipid bilayer bubble, and that's called a vesicle. It's a transport vesicle, and that transports the proteins to the Golgi apparatus. And if this is the membrane of the Golgi, once the transport vesicle touches the Golgi membrane, it can open into and become one with the Golgi. So you know what that means? That means that the transport vesicle actually becomes part of the Golgi itself. It actually fuses to and becomes one with and opens up into the Golgi apparatus. And by the way, do you see this face of the Golgi? This is the side of the Golgi that accepts vesicles from the ER. This is called the cis face of the Golgi. That's the face that's pointing towards the ER, and that's the face that accepts the vesicles from the ER. From there, those proteins make their way toward the other face, the other side of the Golgi, called the trans face of the Golgi. And along the way, those proteins are modified. They're modified and modified and modified. What do I mean by modified? The Golgi can make a number of modifications. Some of those modifications may include being cleaved or cut at a certain spot. Some proteins are not active until they've been cleaved. Okay. Another modification may include some kind of sugar adding on, or myristylation. You don't need to know those terms, but you might add a sugar onto the protein, or you might cleave the protein. These types of events make the protein finalized. So this is called protein processing. So the Golgi accepts proteins, it processes those proteins, and once those proteins reach the trans phase, it then ships those proteins. How? Again, just like the ER pinched and pinched until it pinched off a transport vesicle, the trans phase of the Golgi can pinch and pinch off as what's called a secretory vesicle, and that secretory vesicle is full of proteins, modified proteins, okay? And let me show you something real quick here. Look, so this is what I've been talking to you about. The proteins are originally made at the rough ER membrane that pinches off into a transport vesicle, which fuses to and becomes one with the cis face of the Golgi, delivering the said membrane proteins and other proteins to the Golgi. Those proteins make their way to the trans face of the Golgi, at which point the trans face of the Golgi, the membrane can pinch off into a secretory vesicle, which can fuse to and become one with the plasma membrane, or those vesicles can just float off into the cytoplasm, becoming their own organelles, such as a lysosome or maybe a peroxisome, and just float off into the cytoplasm. So there's two avenues, right? Those proteins can either attach to the membrane. If there are proteins inside of this vesicle, you see these proteins inside, those proteins will be secreted out of the cell. So for example, hormone can get released out of the cell. If those proteins are part of the membrane of this vesicle, then those are going to become your membrane proteins, okay? Whereas these vesicles are going to float off and become other organelles inside of the cell, like the lysosome or peroxisome. And what do I mean by membrane proteins? Let me show you real quick. This is kind of nice to understand, though not fully important for the exam. It's just nice to understand in general. But do you remember what I showed you here? This is a membrane. Okay, and membranes are made of primarily phospholipid bilayers like this, right? Phospholipid bilayer. However, remember I told you earlier there are all kinds of different proteins that exist inside of the membrane, okay? Now, how did all these proteins get into the membrane? Let's say this is the plasma membrane. This is the membrane around the cell. How did all these proteins get into the membrane? Well, exactly what I've been saying. Look, exactly what we've been talking about. How did all those get into the membrane? Those membrane proteins were made here at the rough ER. They pinched off as a transport vesicle. They went to the cis face of the Golgi. They were modified, pinched off at the trans face of the Golgi. That vesicle, secretory vesicle, fused to the plasma membrane, and that's where all those transmembrane proteins came from. That's where all those membrane proteins come from. They were originally made at the rough ER. So just fun facts for you. Okay, so now we know what the Golgi is, hopefully. And by the way, you see these membranous sacs I told you about? These sacs here? They're called cisternae. Cisternae are the membranous sacs of the Golgi apparatus. Okay, so again, the Golgi apparatus is a shipping and processing center for proteins. It accepts proteins from the rough ER. It then ships proteins from its trans side. It accepts the proteins at the cis face, this face here. It then modifies those proteins and ships them from the trans face. Okay? So that's good to understand. Now, I told you that some of those vesicles from the Golgi can become other organelles like the lysosomes or peroxisomes. Those were made originally from proteins at the rough ER, modified by the Golgi, floated off, and became the lysosome or peroxisome. We're not really going to talk about the peroxisomes, but we are going to talk about the lysosomes next. So let's do that. Lysosomes. Okay, so the lysosome was just the organelle I was just showing you, telling you about. Now, lysosomes are basically a vesicle, but you could call it a vacuole, okay, because it's more permanent. It's a vacuole full of digestive enzymes, hydrolytic enzymes. You know what hydrolytic means, right? Enzymes that can do hydrolysis, right? These are enzymes that break down macromolecules. And whenever the cell would like to break down macromolecules such as proteins or polysaccharides or whatnot, it can break them down using the lysosome. Okay, so you could think of it as the digestive organelle of the cell. Okay, and what are vacuoles? Again, I just mentioned the term vacuole. If a vesicle is a smaller, more short-lived phospholipid bilayer ball, a vacuole is usually a larger and more permanent phospholipid bilayer ball. It usually has a function in the cell. You have storage vacuoles. You've heard of storage vacuoles. If you guys remember from lab this week, we did lab 7. Lab 7 covered cells, and we looked at, do you guys remember we looked at potato cells? I'll flash a picture of the potato cells. And do you remember what we did? We sliced the potato razor thin, and then we stained it with iodine. And inside of the crystal-like potato cell, you saw purple dots. And those purple dots are called amyloplasts. The reason they stained purple was because the iodine reacted with starch in the amyloplasts, revealing the starch within the cell. Now, what are those amyloplasts? Those are an example of a storage vacuole, storage for starch. Okay, that's just an example of a vacuole. Another example of a vacuole we talked about earlier when we saw the plant cell, and that's the central vacuole of plant cells. This central vacuole fills with fluid, increasing the turgidity or turgor pressure of the cell. Plant cells want to be high pressure inside, and they want that because the plant wants to grow up straight, grow up strong. And one way to achieve that without having a skeleton like you and I do is by having this pressure inside of the cell. And those cells do not rupture because they have a cell wall. The cell wall keeps them intact. Okay, what else? Contractile vacuoles. Now, this is a really neat one. Contractile vacuoles are neat. Do you guys remember the paramecium we looked at, for example? We saw some of these freshwater protists. Okay, we saw some protists. A lot of these protists don't have a cell wall. So what happens to these creatures is if they find themselves in freshwater, if they find themselves in areas that have higher water concentration, water might, through osmosis, start creeping into the cells. Water will force its way into the cell. And you know what happens if a paramecium or something, if water's coming in, right? It's going to start swelling and swelling. And does it have a cell wall to prevent explosion? No. So those cells might explode, actually. It's a phenomenon called lysis, right? Due to a hypotonic condition, which we're going to learn about in the next chapter, actually. So to prevent explosion, creatures that don't have a cell wall might have what's called a contractile vacuole. It kind of, think about it as a central vacuole that fills with fluid, but then squeezes that fluid. It contracts to squeeze that fluid out of the cell, preventing the cell from exploding or lysing. Okay, so it's pretty an interesting example there. Okay. All right, now we're going to talk about the cytoskeleton, and then we're going to move on and talk about the extracellular parts of the cell. But before that, let's take a quick little break and come back and finish off this chapter strong. And that will conclude the material you need for exam 2. So let's take a little break. Hey everyone, welcome back from break time. Let's do it. Let's finish off chapter 4 so that you have all the material that you need to be successful on exam 2. So chapter 4 finishes off with a couple of concepts. One, let's start with the cytoskeleton. Now, what you need to understand is that cells have a cytoskeleton, just like you and I have a skeleton that gives us structure and support and assists in motility. The cell also has a cytoskeleton that assists it in structure, support, and motility. But it's not made out of bones. The cytoskeleton is, cyto, by the way, means cell, and skeleton, you know what that means, support system. Now, The cell's skeleton is made out of protein fibers. You can see here it says a network of protein fibers. And you can see them in this picture here. This is fluorescence microscopy, which means using a fluorescence microscope. And you can see all this whole network of these fibers I've been telling you about. They span the whole cell, giving structure, support to the cell, also aiding in cell motility. So what are these fibers? Well, there are 3 fibers, okay? There are 3 of these cytoskeletal fibers. One class is called microtubules. Another class of these fibers is called microfilaments. And the third class is called intermediate filaments. So let's talk about all of them. For all of these 3, I want to tell you what they look like, what they're made out of, and what they participate in, what functions in the cell they have. Okay, but before I do, you could see this is one of the let me just show you an example of something before we dive into the actual material. This is one of the fibers called the microtubule, and it's really interesting because microtubules can kind of serve as monorails inside of the cell. So you know what a monorail is? It's a rail on which a train can use as a track. And here, these cellular monorails, these microtubules, can serve as train tracks for cargo vesicles. You remember cargo vesicles, like the transport vesicles from the rough ER? Well, they can make their way to different parts of the cell. How would they go? How do these vesicles travel? Well, there are these motor proteins, literally proteins that are motorized with the help of ATP. These proteins can walk, literally walk, along the monorail, along the microtubule. So imagine, actually, here's a electron micrograph of it here. This is a real picture of a microtubule, and here you have your walking legs. Okay. Let's see. Let me show you something. Do you remember this? Do you remember when I said the vesicle leaves the rough ER and it makes its way to the cis-phase of the Golgi? Well, how does it do that? Well, one way would be if it attached itself to those motor proteins and then walked along microtubules till it reached the cis-phase of the Golgi apparatus. It's pretty interesting stuff. Anyway, that's just one neat example before we dive right in. So let's dive right in. Excuse me. So see this actually here. Sorry, I got the hiccups. See this right here? This is a good reference slide, actually. If you want to keep referring back to this slide, it will help you to understand the difference between the 3 different fibers. So let's jump right in, and you'll see why. In this first column, which I'm highlighting, in this first column, you have talk of the microtubules. In the middle column, it talks about the microfilaments, and in the third column, it talks about the intermediate filaments. So let's start with the first column here. You see, let's talk about the microtubules. These microtubules are the thickest. What you need to understand is these microtubules are the thickest. Look how the diameter is much thicker than the other 2. It's the thickest cytoskeletal component. Additionally, look what it looks like. Look at its design. Look at this cartoon. It looks like a hollow tube, doesn't it? Microtubules look like a hollow tube, like a drinking straw. And what are they made out of? What are these hollow tubes made out of? If you look at the cartoon at the bottom left, you'll see that the hollow tube itself is actually made out of thousands and thousands and thousands of these proteins. Each one of these little dots is a protein. There's 2 main types of protein. There's alpha and beta tubulin. Okay, alpha tubulin and beta tubulin. So you've got what are known as alpha beta tubulin dimers. And then the microtubule is made out of hundreds and thousands of these dimers. Okay, so do you remember what a quaternary structure is? It's when numerous tertiary structures, numerous proteins come together to form a structure or function greater than itself. This would be A microtubule would be an example of a quaternary structure. In fact, the alpha and beta tubulin is also a quaternary structure right there. So, what have we learned so far? Microtubules are the thickest of the cytoskeletal components. They look like hollow tubes, and they're made of proteins called alpha and beta tubulin. But what do they do? What do they do? I already told you one function. The microtubules serve as monorails inside of the cell for transporting things with motor proteins. That's kind of neat. But let's look here. There's other functions here. You see what I'm highlighting here, what I'm actually circling? These are the functions of the microtubules. Okay, so they can maintain cell shape. They can assist in all these things. So let's talk about stuff. Let's talk about some of these things. Look, here's an example of something microtubules do. These are microtubules right here. Okay, and not only is it a microtubule, it's actually, this looks like a triplet of microtubules next to a triplet of microtubules next to a triplet of microtubules. So it's like a ring of triplets. And this is a bundle of microtubules, and you can find them in animal cells. Does this look familiar from before, this bundle of tubes? Weren't these the centrioles? Remember centrioles? Centrioles are made of microtubules. Do you remember this? Let me go back real quick. Here, remember the animal cell? Do you remember this structure I told you to understand? This is called the centrioles. It's two bundles of ring of triplet microtubules. And do you remember what I said? Animal cells have centrioles. Plant cells do not have centrioles. And the centrioles, we're going to talk a lot about these in a later chapter. I believe chapter 10, when we talk about mitosis, when the cell is dividing. So when the cell is dividing, the centrioles play a big role, and they play a role in separating the chromosomes. Okay, so that's all you need to know. What do we need to know? We need to know that here. We need to know that the centrioles in the animal cells are made out of microtubules, and they play a role in chromosome separation when the cell is dividing. So just know that. Another very important thing that, or function I should say, another very important function of the microtubules is they play a big role in cell motility of eukaryotic cells. They play a role in flagella. Like, you know, your sperm cells. Sperm cells are flagellated. This is an actual picture of a sperm cell on the right. You've got the head part of the sperm, which contains the genetic material, but then you've got this long whip-like structure. That's called the flagella. And if you have any idea about how sperm move, they have this flagella that beats like a fish tail. It beats back and forth, back and forth, back and forth, and that propels the sperm cell forward. Okay, what I want you to know is that microtubules are essential for flagella function. In fact, the flagella is lined with a bunch of microtubules. I'm going to show you exactly what the microtubules look like inside of the flagella in a minute. And another type of structure that depends on these microtubules are called cilia. Cilia are short hairs. These are short hairs that beat. They're much shorter than a flagella. Flagella is like a long whip, all right? In comparison, cilia are short hairs. And usually, a cell would be surrounded with cilia, many cilia, whereas usually a eukaryotic cell only has one flagella, right? And where do we see cilia? Because we know we see flagella with sperm. Where do we see cilia? Well, do you guys remember the paramecium cells? I'll show you a picture of that. The paramecium cells from Lab 7, those were being propelled, paramecium were being propelled by cilia, the beating of the cilia. Okay, where else do we see cilia? This is fun fact for you. The lining of your lungs, inside of your lungs, you have cilia. and these cilia beat. Okay, and it actually, this is a side note. You don't really need to know this for the exam, but it's a fun thing to understand. The inside of your lungs is coated with cilia. These cilia beat. Okay, these cilia beat in unison, and they beat in one direction. So let's say you're out and about. You're breathing. You're breathing in dust. You're breathing in mold, and that dust and mold settles inside of your lungs. You really don't want dust and mold settling inside of your lungs, do you? So your body has a mechanism for bringing that dust, bringing that mold back out of your lungs so that it can make its way to your esophagus, down into your stomach, where it can destroy those germs, those potential harmful things. So what happens is, it's called a mucociliary blanket, mucus cilia blanket. When you breathe in, the dust settles in the lung, but these hairs are beating in one direction. And because they're beating, the dust kind of just moves along out of the lung, right? And the mucus is also pushed with it. So this is a very, very good point about another really good reason to not smoke. You should definitely consider not smoking because it does cause cancer. However, another really good reason not to smoke is because you can damage those cilia and that mechanism, the mucociliary blanket mechanism of your lungs, the mechanism that's designed to clear your lungs of debris. You can actually damage that mechanism and damage those little hairs that are trying to keep your airways clean by smoking and covering them in tar and other chemicals. Okay, and that's why it's more likely to get lung infections and such if you are a smoker. So anyway, that was a fun little fact for you. So another thing I want you to know before I tell you exactly how the microtubules work in the flagella and the cilia, another thing I want you to know is the difference between how the flagella beat and how the cilia beat. Okay, look at the flagella. It's like a fish tail. It just beats back and forth, back and forth, back and forth, equally, back, forth, back, forth. All right, so kind of like a fish tail. Okay, whereas the cilia, what I want you to know about the cilia is that it's not just an equal back and forth motion. It's not just back forth. Notice what it says here at the bottom. Notice what it says here. There is a power stroke. So it goes woof, and then it like recovers. So can you imagine like the breaststroke when you're swimming? You do a stroke, and then you kind of bring your arms in, you recover, and then you do another big stroke. So that's a lot different than going straight back, straight forth. Okay, so I want you to know that flagella beat back and forth like a fishtail, whereas cilia, power stroke, recovery. Okay. So let's talk about, let me talk about the structure of the flagella. Imagine if we could look at this flagella. Imagine if we could look inside the flagella. What if I could look inside this flagella right here where my laser is? Look inside. So let's look at that. That's what this is. Imagine if I could peel the skin back, the plasma membrane back, because the flagella is coated with plasma membrane. Imagine if I could peel that back and look inside of the flagella. That's what this is right here on the right. If I could see inside the flagella, you see what I'm saying? All of these microtubules. See, these are all microtubules. And these are doublet microtubules. You know what that means? Double microtubules. Look, how many doublets are there? Here's a doublet. 1, 2, 3, 4, 5, 6, 7, 8, 9. So what they say is there's 9 doublet microtubules in a circle, and how many microtubules in the center? There's 2 single microtubules in the center. So you know what they call this? They call this the 9 plus 2 arrangement of microtubules. You know why? Because you have 9 doublets encircling 2 singlet microtubules. Okay, it's the 9 plus 2 arrangement. So why does this move? Why does this facilitate motility? Well, look at this. Here you have. Let's zoom in on this part here. Let's zoom in on this part that I'm circling here. Let's zoom in on that. So you got, a doublet microtubule here and a doublet microtubule next to it. Well, look what you have between the doublets. Look what you have. You see what I'm showing you right here with the laser? What are these little red Y-shaped things? Do they look familiar? These little red Y-shaped things, do they look familiar at all? Yeah, do you remember when look. Do you remember when I introduced the microtubules? I said microtubules can serve as monorails for these little motors, and the motors can actually walk on the monorail on the microtubules. Well, that's kind of what these are. Look, these little guys, these little red-shaped things are motor molecules called dynein proteins, and they're little motors. With the help of ATP, they literally march. Okay, so let's look at these two. Let's look at these two here. So if you have a doublet microtubule and a doublet microtubule, okay, what happens if I provide this with ATP? What happens if I provide this with ATP? Well, these little feet start to march, and they all march in one direction, and that causes this whole thing. These little feet, because their heads are attached to the doublet on the right, their marching feet are attached to the doublet on the left. So with the help of ATP, all the little marching legs march like a millipede, and that bends the entire structure to the right. You see, so basically that's how the motility works in this flagella and the cilia. You've got doublet microtubules with motor molecules called dynein in between. With the help of ATP, that marching, that motion, that motility in the motor molecules causes a bend in the microtubules, hence the bending of the flagella and the cilia, and hence the motility. So that's kind of how they work. You don't need to know it in that much detail, but you might want to know what dynein is. You know that there's a 9+2 arrangement of microtubules, and that they're responsible for flagellar and cilia motility, and that flagella beat back and forth evenly, whereas cilia do a power stroke. All of these things are nice to know. Okay, so that's kind of cool, isn't it? So microtubules, without microtubules, you couldn't have things like flagellar or ciliary motion. And that's pretty much all I need you to know about the microtubules, okay? So again, real quick summary. Microtubules look like this. They are the thickest of the cytoskeletal components. They look like hollow straws or hollow tubes. They are made of alpha and beta tubulin, several of them, right? Thousands of them. And they are involved in these things here, these processes here. They are involved in formation of the centrioles, and the centrioles do what? You should know they play a role in separation of chromosomes during cell division. They are involved in the flagella and the cilia, and the flagella beat back and forth. The cilia have a power stroke. And there's a 9+2 arrangement of microtubules in the flagella, and with the help of dynein, there's motility. Okay, so that's pretty much it. So now let's move on to the microfilaments. Let's go on to this column here, the microfilaments. Okay, these are also called actin filaments. Why? Because let's take a look. First of all, what does it look like? Look at this. It's the thinnest of the cytoskeletal components. It's got the smallest diameter. And if you look really closely, what does it look like? If you look at it really closely, it looks like two pearl necklaces. Imagine two pearl necklaces that you've twisted or intertwined, right? You've twisted the two pearl necklaces together. And that's what microfilaments look like, and they're the thinnest of the cytoskeletal components. Okay, now you see each of these pearls in the pearl necklace. See each of these pearls in the pearl necklace. Each pearl then is a protein called actin. Does that make sense? So again, this is an example of a quaternary structure of thousands and thousands and thousands of actin proteins, right? Actin proteins make up the actin filaments, also known as microfilaments. So again, you can call them actin filaments, you can call them microfilaments, sometimes they call them actin microfilaments. But that's what they are. They're very thin, Looks like two pearl necklaces, and it's made up of a bunch of actin subunits, actin proteins, okay? So what are the jobs? Now that we know what it looks like and what it's made out of, what are the jobs of the microfilaments, that kind of cytoskeletal class? What do they do? So they do these jobs here. Again, this is a good review here. These are the jobs, but let's go through those jobs one by one. First of all, the microfilaments form a three-dimensional network, a 3D network called the cortex. Okay, you should know that they form a 3D network called the cortex just inside the plasma membrane. So right underneath the plasma membrane. If this is the plasma membrane, they're right under the plasma membrane. Okay, and why is that important? Well, because if the plasma membrane is, loose material like a tarp, well, whatever's right below the plasma membrane is going to give shape to the tarp, right? So like this, look, if this tissue is the plasma membrane, it doesn't have any shape to it, right? This tissue doesn't have any shape, but whatever is going on underneath the tissue is going to give shape to the tissue, kind of like a tent, right? Think about a tent. You ever gone camping? Well, you know that a tent has a canvas, right? It has a canvas material, and then it has, poles that you put into the tent, right? You put a tent poles in the tent, and because of the poles in the tent, that's why the tent has a shape. So what you need to understand is, what I'm trying to say is that the canvas material, the clothy material of the tent is like your plasma membrane. It doesn't really have a shape. Now, it's the poles that give it shape, and the poles are what I'm trying to say are the cortex or the microfilaments. So if the microfilaments do something under the plasma membrane, the cell's going to look like that as a whole from outside, if that makes any sense. So let me explain. Let me show you an example. We looked at this example in the lab this week, didn't we? We looked at lab 7 cells, and we looked at those amoeba cells. And do you remember amoeba are these blob-like creatures that move around really slow, but they move by formation of what are called pseudopods. What does that mean? A portion of their plasma membrane extends as a pseudopod or a fake foot. Okay, and that's how they move about, and that's how they get to food, and that's what they do. And part of their plasma membrane, part of their cell membrane might retract, and another part may extend, right? How is it negotiating this? How is it doing that? Well, it's rearranging its cortex, right? It's just like if you have poles and you're inside of a tent. If you build a long pole, if you're making a long pole, you're going to push out that part of the tent. If you're breaking down a pole, that part of the tent is going to retract. Does that make sense? So how does a amoeba move around? Well, the leading parts you see here, the parts that are making the pseudopods or fake feet, they're building actin microfilaments. They're building more and more poles. The parts that are retracting, the parts of the amoeba that are coming back in or flattening out, they're breaking down the microfilaments. They're breaking down the poles. Okay, so that's kind of interesting to understand. Here's another example. This is an example in humans, right? You and me. Our intestines have a lining. The intestinal lining has a very interesting structure to it. The cells of our intestines have finger-like projections on them, okay? So the cell looks like your hand, and then part of the cell looks like fingers, right? Finger-like projections. You can see them here, these finger-like projections on the cells of the intestines. Why would you think that the intestinal cells would have finger-like projections? And by the way, the fingers are pointing toward the inside lumen, the inside part of the intestines. You know where that is? That's the part of the intestine where the food and the drink and what you've eaten and drank, where that food is passing over the fingers. Why do you think those cells would like to have finger-like projections? Well, because the finger-like projections increase the surface area for absorbing nutrients, right, from the food and from the drink. Okay? So, but what's allowing the cells to have these finger-like projections? Well, look. Here's the finger-like projection of plasma membrane, right? And what's right underneath? What's holding it up as a finger? Microfilaments. Microfilaments. The microfilaments of the cortex are allowing these finger-like projections to form. And what you need to know is that the finger-like projections of these cells in your intestines are called microvilli. Microvilli. Microvilli are the finger-like projections of your intestinal cells. And what's allowing that to happen? The microfilaments underneath in the cortex, holding them up in that fashion. But unlike in a pseudopod, once you form those microvilli, you don't need to retract them and make more and retract them and make more. You make them and leave them. Okay, so that's pretty interesting stuff. Okay, here's another reason microfilaments are so cool. Look at microfilaments, okay? Your muscles would not be functional, would not be possible without microfilaments. You know, your muscles would not function without microfilaments. And here's why. Let's look inside. Your muscle cells are actually fibrous. They're long, okay? And if you were to look inside of a muscle cell, this is what you would see, okay? This is what you would see inside of your muscle cell. So this here is one wall of the fiber of the cell, and this here would be the other wall of the cell. And look what's attached to the walls. You see these orange structures? These are the microfilaments. Okay, so before I get into that too much, do you guys know why your muscles work? Do you know why your muscles work? So my bicep is working right now. Okay, my bicep is working right now. Do you know the difference between what's going on when my arm is extended and what happened when my arm has contracted? Well, the term contracted should mean something. The bicep contracts. It actually literally becomes shorter, right? It contracts. And that pulls on the bones of my forearm, the radius and the ulna. It pulls them close to my body, right? Because the muscle contracted. The muscle got shorter. Why do my muscles get shorter? Well, it has everything to do with microfilaments. Now imagine this, okay? Imagine this with me. You guys ready for this? Oh gosh, do I have an example here on hand? I might actually. Hold on, let me get an example. Let me get an example and I'll come right back. All right, I'm back, everyone. So I was going to give you an example of what's going on inside of a muscle cell. Now, just to give you an idea of how important the microfilaments are inside of a muscle cell, let's say that we are inside of a muscle cell right now. Okay, I am inside of a muscle cell right now. Okay, so the square you see around me, you see this here? This is a cell. We're inside of a cell. Now let's say on one wall we have a rope attached to the wall. You see, so this rope is attached to that wall, okay? And on the other wall we have another rope attached, okay? This rope is attached to this wall. See, I've got a rope attached to this wall. I've got a rope attached to the other wall. Okay, so this is basically a muscle cell. Okay, and do you know what the ropes represent? These ropes that are attached to the walls. These ropes that are attached to the walls of the muscle cell are basically the microfilaments. Okay, you see there in your book, it's the orange lines I was just talking about. So again, if you're inside of a muscle cell, the ropes attached to the walls are the microfilaments. So what would happen if I were to go ahead and pull these ropes together? Right? What do you think would happen if I pull the ropes towards one another? And the ropes are attached to the walls. Let's try it. Let's try it. Okay, I'm going to try it. Oh man, this is going to be scary. I'm going to try it though, alright? Okay, let's do it. Let's pull the ropes. Okay, so what happened there? I don't know what happened there. I don't know if I have the editing skills to show you what happened there, but hopefully I did and do. But you saw that when I pulled the ropes, the walls came in. So look at this. Inside of, let me show you here. Inside of your cells, inside of your muscle cells, you've got the microfilaments, those actin microfilaments attached to the walls. Now, remember what I did? I pulled those in. I pulled those ropes towards one another. But you know what represents me in this case? This purple stuff. What is this purple stuff? It's called myosin. It's a motor protein, okay? Imagine if I had a stick, okay? Imagine if I had a stick and that stick had walking legs on it, okay? And this side of the stick liked to walk in one direction, you know, this side of the stick likes to walk in one direction, you know, pulling the rope towards this way, and the other side of the stick likes to walk in the other direction, pulling the rope the other way. Does that make sense? I have a motorized stick with walking legs going in opposite directions. That's essentially what this purple stuff is. This is myosin. So the reason why your arm contracts, and comes in is because myosin motor proteins, they have little walking heads, and they're walking in opposite directions, pulling the ropes together. And that causes the cell to shorten, that causes the muscle to contract. So anyway, I hope that was a fun little demonstration of how the muscle cells contract inside of your body. It's all because of actin and myosin. So without that actin, the microfilaments, you wouldn't have muscles. That's what I need you to know. Another cool thing, you may have seen this in the lab when we looked at the allodia cells. You may have seen that if you look close enough, you could see that the little chloroplasts were floating around the allodia cell. And of course, this is the allodia cell. It's a brick-shaped cell. And the chloroplasts were kind of moving around, almost like someone flushed the toilet, and they're just going around and around in a circle. That's a phenomenon called cytoplasmic streaming. It's a circular flow of cytoplasm within the cells, and this helps to speed distribution of materials within the cell. It just gets materials to where they need to go around the cell. And what I need you to know is that microfilaments are central to this process. You don't need to know about the sol gel transformations that drive this, but just know that without microfilaments, this cytoplasmic streaming phenomenon would not be possible. And the last thing I want you to know about the microfilaments is that they are responsible for this cleavage furrow formation in animal cells. What does that mean? When an animal cell wants to divide into two animal cells, okay, when an animal cell wants to divide into two, you see what has to happen? The plasma membrane has to pinch until you've got two new cells, right? That pinching in animal cells, that pinching is called cleavage furrow formation, and that is dependent on microfilaments. Without microfilaments, you wouldn't have cleavage furrow formation, you wouldn't have cytokinesis, which is cell division, or the separation of the cytoplasm like we know it. Okay, so again, what do you need to know? That cleavage furrow formation is dependent on microfilaments. Okay, and then let's talk about intermediate filaments. This is the last type of filament. Let me show you here. Yeah, so we've talked about microtubules. They were the thickest cytoskeletal component. They look like big hollow tubes. We talked about microfilaments. They look like two pearl necklaces that were intertwined. Those are the thinnest. And now we're talking about the intermediate filaments. These are intermediate or medium in diameter. They're in between in diameter, and they're usually made of this protein keratin. Okay, these proteins look like kind of like a power cord, right? They're all twisted together. Okay, they're coiled together. And what I need you to know about these intermediate filaments is that they do a lot of things, but they don't do anything involved in motility. You know how the microtubules were involved in flagella and cilia, motility, and the microfilaments were involved in pseudopods and muscle contraction, all this cool stuff. Well, the intermediate filaments are not involved in any of that. The intermediate filaments are mainly for anchoring things in place, anchoring things like the mitochondria in place, organelles in place, giving structure, support, anchorage to things. Okay, that's pretty much it. Now, one thing I do want you to know about the intermediate filaments is that the intermediate filaments make up the nuclear lamina. Does this term look familiar? It should, because, let me show you. Let's flip back. Let me show you. Let's flip back. Look here. Remember when we were talking about the nucleus? I told you at the bottom that the nuclear lamina is a structure in the nucleus, around the nucleus actually, that gives structure, support. It basically maintains the shape of the nucleus. That's called the nuclear lamina. And now we know, what is the nuclear lamina made out of? It's made out of intermediate filament. Okay, so it's made out of intermediate filaments. So that's good to understand. Let's please understand that. Okay, so if we look at this summary slide now, let's look at the summary slide. Look at the summary slide. You can see a cell. In orange, we have the intermediate filaments. What are the orange intermediate filaments doing? It looks like they're just anchoring things in place, kind of like a network of anchoring, like a scaffold. What are the actin microfilaments doing? They're right underneath the plasma membrane, the cell membrane, in the cortex, giving shape to the cell. What are the microtubules doing? They're basically monorails that span throughout the cell. Okay, so now we know something about each of those, and we've pretty much addressed the cytoskeletal components of the cell. There's only one more concept to this chapter, and then we're done with the material for chapter 2, and that is extracellular components. These are components outside of cells. So let's talk about extracellular components. By the way, extra means outside of. Cellular obviously means the cell. So these would be extracellular components, would be structures outside of the cell, right? For example, what do we know in plants? What kind of structure do we have outside of the plasma membrane, outside of the cell? We have a cell wall, right? Plants have cell walls. And we talked about this in chapter 3. We talked about cellulose and how cellulose is made of beta glucose. Remember all that? And what you need to know is that cellulose makes up the cell wall of plants and some of the protists. Okay? Whereas fungi also have a cell wall. Fungi are eukaryotic, remember, like mold and yeast. Fungi also have a cell wall. That cell wall is made of chitin, which is also based on glucose, but you don't really need to know that. Just know that fungi have a cell wall of chitin. Plants and some of the protists have a cell wall of cellulose. Okay, but do animals have a cell wall? Do you guys remember? Do animals have a cell wall? Absolutely not. Animals do not have a cell wall. But does that mean we have nothing? Here's the thing. All right, so animal cells do not have a cell wall, but they do have something. They have some kind of support system, and that support system is called the extracellular matrix. All right, animal cells have an extracellular matrix, also known as the ECM. And the main component of the ECM, you can see it here in this picture on the left, the main component of the ECM, because this right here is the plasma membrane. You see this? This would be the membrane of the cell. The main component outside would be the collagen fibers. You may know the word collagen. You know beauty products, skin creams and such. They advertise now with collagen, with extra collagen. What they're trying to do is increase the amount of this collagen protein in the cream, because collagen gives structure and support to your cells. In fact, as you age, that ability for your skin cells to maintain the collagen, to maintain the ECM, the extracellular matrix, is degraded over time, and that contributes to wrinkles, that contributes to sagging, droopy skin with age, right? So that's why beauty products try to reincorporate that in there. How effective they are, I'm not sure. It's up for science to decide. But here's here's what the ECM is made out of. It's made out of collagen fibers, which are basically proteins. It's a quaternary structure of collagen proteins. And it's also made out of this barbed wire looking stuff. You see this stuff here? It looks like barbed wires. That there is called the proteoglycan complex. Proteoglycan complex, you don't need to know much about it. You need to know that it's part of the ECM, of course. But it's made out of, it's really interesting because the main part of it's made out of sugar. Okay, the branches are made out of protein. And then the twigs off the branches are made out of sugar again. So it's like sugar, protein, sugar. It's like a tree made out of sugar, protein, sugar. It's weird. So again, what are the main parts of the ECM? Collagen fibers made out of protein and proteoglycan complex, which is like this combination of protein and sugar. And all of this coats your cell, around your cell. It's not as strong as a cell wall, but it is something. It does give elasticity and strength to your cells to some extent. Okay. And what else do you need to know? You need to know that the collagen and the fibronectin attach to these little adapters, these little molecules called fibronectin. And the fibronectin attaches to these anchor proteins. You see these transmembrane anchor proteins called integrin. And integrin not only connects to the fibronectin and the collagen and the proteoglycan outside, it's also connected to or anchored to the microfilaments in the cortex inside. Remember, underneath the surface, underneath the plasma membrane, you have the cortex, which is made up of the cytoskeletal component microfilaments, actin microfilaments. Those are connected to integrin, which is connected to fibronectin, which is also connected to collagen and proteoglycan complex. So the way I remember it is microfilaments are attached to integrin, integrin is attached to fibronectin, and the fibronectin is attached to the collagen and the proteoglycan complex. So basically that way you're anchoring the inside of the cell to the plasma membrane to the outside of the cell, and that really does give some structure and stability and elasticity to your skin. So is it as strong as a cell wall? Nope. Is it better than nothing? Yep. So that's that. That's the extracellular components or component of the animal cell. And that does lead us to the end of the chapter. So that's it. That's the material for exam 2. Thanks for bearing with me. Thanks for listening. I hope you got a lot out of that. I hope you're prepared for exam 2. Remember, exam 2 covers only chapters 6 and 4. I do have a review video up that you can watch and learn from for the exam coming up. Look under test reviews, and you'll be just fine. Study hard, and thank you for watching. I hope this all helped. All right, and good luck on your exams. Bye bye.